Dr. Mayers, the floor is yours.
Great, thank you very much. It's really a pleasure to be asked to chair this panel of distinguished experts. And the topic of the discussion is driving the next green revolution. So how can we address the challenges that are faced by the rice community? How can we continue to maintain the improvements in rice yield? And what can really, as genome scientists, what can we contribute to the continued improvement of rice and overcoming the obstacles which we face? And so what I thought I would do is ask each of the panelists to just speak for a couple minutes about what they see the major challenges are from their perspective with their expertise. So if we could work our way across, maybe we can start with Jinan at the end, and then we can open up the floor. And so this is really meant to be an open discussion, an opportunity for give and take. And so I really strongly encourage you, the members of the audience, to participate in this. So it's not just a bunch of talking heads up here at the front. We really want to hear from you. We want to be able to address your questions. If we can't address them, we want others in the audience to address them. It's really a good opportunity for some back and forth on this. So maybe if we could start, Jean, would you be willing to say, just a quick summary of what from your perspective are some of the major obstacles that are facing us?
Good afternoon. I hope you are having a good time. Anyway, I have been involved in making tDNA population, and we are distributing over 30,000 mutant line every year throughout the world. I know that there are big demand, and we hope to continue to do this service. Unfortunately, ours and others all together, as the morning session speaker mentioned, that it covers about 50%, maybe less, considering some of the information may be not correct. So with this, of course, we need more. Ideally, there are speakers calculate about percent over 95% frequency, but ideally we like to have at least 2 mutants per each gene. Therefore, you don't have to spend a lot of time making RNAi or whatever. Unlike Arabidopsis, it takes at least a year or 2. If you are looking for recessive mutant, easily 2 years to confirm the phenotype. And since tDNA or ACDS has high background, often your phenotype you observe is not correct. So after 2 years time of RNAi, if it's not confirmed, then your effort is wasted. Therefore, how we can make more mutant and available, that's a big challenge. I don't know answer. Let me just add one more. Why scientists like me or I don't know, Chip, how many he's making more, or Caroline, why we are making more? You guys, you may say that you have only the expertise and all those things. Problem is there's no challenge, no reward. In other words, say I make 50,000 more mutant in certain variety and then submit to Plant Journal or Plant Cell. Will you recommend accepting that paper, or will you reject it? I almost think that it's likely be rejected it. So without publication, I cannot convince my government, oh, please give me more money. So this is a big dilemma. In other words, those people who already has experience and so on, how they get reward. I'm not saying that, money or whatever, the acknowledgement, whatever. How can you get the reward so that we can go back to the government that this is worth doing it? I think I'll stop over here. I'd like to hear about your answer for that. Thank you.
Thank you very much. Val?
But as you may have heard, I've been involved with rice genome research for the last 20 years. And anyway, as far as my institute is concerned, Since the completion of the rice genome sequencing project, we've focused mainly on functional characterization of the genome, and we had projects actually on analysis of genes relevant to agriculturally important traits in rice. And with various approaches, we were actually able to elucidate the molecular network of many agronomically important genes. So we have also developed resources which I think the community is using right now, the full-length cDNA clones, the TOS17 lines, also genetic analysis materials. And we're now involved in projects which aim to attach function to each and every annotated gene in the rice genome. And in the concurrent sessions tomorrow and on Wednesday, we'll be describing some of our efforts on characterization of the gene function. So, in addition to all these initiatives that we've been doing, like the sequencing, core collections, wild relatives, development of resources, and global analysis of the transcriptome, I just want to say that maybe. Okay. I just want to say that maybe there's one area that the rice research community is not addressing at the moment. And that is the question of how can we top the rapidly emerging paradigms of epigenetics and epigenomics to understand deeply the foundation of trait transmission and expression in rice. So I think that the rice research community is not really bent on looking deeper into the roles of epigenetic marks, for example, in regulating gene expression. But it's very clear from the emerging data that if we are to be successful in mining the germplasm for superior mechanisms, for example, related to an expression of yield potential, for example, or heterosis, or hybrid vigor, or transgressive variation, or even the C3C for photosynthesis, then we really have to start looking into the processes like DNA methylation and chromatin remodeling. Because although we already have the sequence of thousands and thousands of rice cultivars, it seems that genomics and genetics alone would not make us jump into achieving the so-called yield frontier, which is the ultimate goal of rice projects. It seems that DNA sequence and DNA variation is only part of the story, and I strongly believe that, based on current information, that DNA methylation-mediated gene regulation is the missing piece of the puzzle. So, what I want to say here is that there has to be an agreement in the community that the next layer of the onion that needs to be peeled in the post-genome sequencing era is the complexity brought about by differential methylation of the DNA and how that affects the expression of genes. But I think one major problem here is that I think epigenetics has always been considered as a special case, or as a rare developmental mechanism in plants. But I think that in reality, these processes it's an important layer of gene regulation per se, and is involved in any phenotypic variation that is of agronomic importance. So it's mentioned here in the old version of RICE 2020 that something mentioned here about systems-wide epigenomes, and I think this is really one focus of research that we should deal on in the next 5 years, in conjunction with all the projects on integrating the databases, developing more resources, because these may be the key to really understanding the complexity of the genome. So I just want to say that we can probably consider the genome sequence as a complete sentence with the subject and the predicate. But if you put methylation into the equation, then what we're doing is actually we complete the whole story. Like we're adding additional modifiers. We're adding more what do you call this? Adjectives or adverbs. And I hope that Blake would support me in this based on your experience in Arabidopsis, for example, and your many works on DNA methylation. So this is one area of research that I just want to lay this table as part of our efforts, or the next in the next 5 or 10 years for rice genomics. So.
Thank you. Could you shut off the microphone, Al? Okay, Eero, thank you.
Thank you very much, Mr. Chairman. To start with, I perhaps need to introduce my background and my view to this issue of producing more rice. I'm plant breeder, coming from private breeding industry where I have been working the last 20 years, not with rice but with other crops. Temperate cereals, oil seeds, in Northern Europe, North America, completely different part of the world. And certainly the view how I see what we need to do to really to respond to the big demand and to drive the next generation of green revolution is kind of reflected of that experience. To my view, if you look at how we are breeding rice today, we are quite a bit behind on what is happening there with the other crops like soy, corn, wheat, barley. Basically where the private breeding industries, very big companies, has been contributing to improvement of these crops quite some time, and of course with quite a big resources. And what has happened there is that quite a tremendous genetic gain and improvements, just making things a little bit better than so-called conventional breeding, and basically using the genomic information to improve the old equations, if I would use this word, of the plant breeders. Of course, we need to look at the full question, so to say, and really to see what we can do, applying the full range of genomic tools. But my view to this is that to respond quickly, as we need to do, the first thing to do is to do the basic things right. Also, which is good that, no matter is it a breeder or geneticist or genomic people, we tend to see that we need to do everything. But I think that the next phase of green revolution will happen in the same way as the previous one. Genetics, breeding, genomics is one part of the story. And the other part is the production systems which are out there. Like, for instance, if we look at the situation in East Africa, it's not the varieties or the genetic part which is the bottleneck. The good varieties are already there, which can produce many, many times more than the traditional varieties which are out there. 2-meter tall land races producing some 1 ton per hectare. And there are already varieties which with a good management can produce 7 ton per hectare. The issue is the production system, the lack of investments. The country has a lot of water, a lot of land. What is lacking there is really the infrastructure and know-how. So I think that obviously, plant breeders tends to be pretty pragmatic. So no question we need to look at the long-term developments and really how genomics can help us as an extra tool to speed up the process. But I would say that the most immediate action we need to do is to really to do the same as what has happened in the other, what I say, industrial crops, and to use the information you genomic community are making for rice available for the breeders. I think I stop here. Thank you.
Great. Thank you very much. Joe, would you like to present your viewpoint?
Okay. So I wanted to comment on do I think it would be beneficial to have a C4 system in rice, which is what Paul Quick was addressing in his presentation today. To me, it's evident that it would be beneficial. I think it would be beneficial if we had it right now, and also look into the future in terms of climate. If we have warmer temperatures than we have now in some areas, if we have more limitations on water, those are conditions where CO2 becomes more and more limiting in terms of getting into the leaf and getting to the rubisco and the C3 cycle. And I think there should be with one is what should be the model for doing that, and Paul has presented the Kranz type system, and I think there are other alternatives. And one is, how should it be done? Is there the will to do it, and are there the resources to do that? So one is the Kranz system that Paul talked about, where you have two photosynthetic cell types, and that is a long-term goal that Paul discussed. There's the biochemistry that's needed for the C4 cycle, and as Paul was describing, the veins need to be closer together, and also the bundle sheath cells need to have a lot more chloroplast, and then the differentiation of the chloroplast function between the two cells. And we know a lot about the biochemistry that's required, the genes required for that, and there's more information coming out about transporters that are needed, like to shuttle metabolites between the chloroplast and the cytosol. But what we don't know, the structural genes, closer veins, and getting the bundle sheath with the chloroplast. So the alternative is what we call a single cell type C4 system, where it would function. Well, one of the simplified ways of that system is no structural changes needed in rice, where the system would be functioning in the existing photosynthetic mesophyll cells. And the enzyme that Paul described, pepcarboxylase, in all of C4, it's capturing CO2 in the cytosol outside the chloroplast. And so in a single cell system, it would capture CO2, make the C4 acids, and then the C4 acids, they decarboxylase, release the CO2, where Rubisco can utilize that. And there's been some work over the years, that's not been a team effort like we have with that's being administered now through ERIE. But there's been, some genes that have been put in to rice, like pepcarboxylase. But there's no complete system that's been put into, the single cell. And I think some of the same constructs that are being made now for putting into rice with the idea of Kranz could be directed or modified towards developing this single cell system. And I'll talk a little more Wednesday about single cell C4 photosynthesis. But either direction, I think there's potential as it progresses to get what we would call a C3, C4 intermediate. There are species that naturally occur this way, and that in itself could be beneficial, whether it's Kranz type or C3 type. also, just to mention, besides C4, I think there are other strategies. With rising CO2, if over the next years, if it continues to rise, then it may be beneficial to put a C4 type rubisco into rice and replace the C3 form, because the C4 rubisco has kinetic properties where it functions more efficiently if you have higher CO2 available to the enzyme. And then other things could be considered downstream, like increasing the capacity for starch biosynthesis and so forth. So I think there are a number of strategies which could be considered for the future.
Great, thank you very much, Gerald. Ben?
Okay, I think I'm one of the persons to be involved to propose the RISE 2020 strategy a few years ago. So I think if we really wanted to promote to work together for this strategy, we should set up we should focus on some area, because for RISE functional genomics project, the research is so diversity, and many years ago, as Rod mentioned, when we work on the RISE genome sequence project, the target is clearly, and also we can share the information, share the back library, share the clones, everything looks easily to work together. But for the functional genomic study, to share the materials, to share the germplasm is more difficult. So I would like to suggest that in this conference, we should set up a rule or regulation for exchanging the germplasm. But for my group's researchers, I feel very lucky, because I have collaborated with Nori Kurata in Japan, and also I have closely collaborated with other Chinese groups, so I can get the germplasm easily. But for the RISE society community to share this germplasm looks very difficult, because they should have some agreement to sign, between the different countries, then to can ship, because most of the germplasm are elite variety. They cannot be freely, to deliver, to distribute to other lab. So this is the first thing I would like to suggest. That if we really want to work together, we should set up some regulation or rule to share the germplasm, RISE germplasm. Secondly, I would like to suggest that if we, for RISE 2020, we should have some people working together to integrate all the published data, then to make this data available and easily to be accessed to use for the functional analysis. Because in my lab, we found when we work on the genome-wide association study, we identify some candidate region. But if we integrated some published profiling data, we also can associate some genetic variation or the profiling data to narrow down the mapping region. So I think if in this society, if some lab are interested in to work on this work, then to update the website, like Blake talked earlier, people working for the rhabdosis project. So this is the second question. The third one, I feel, if we really to convince the government, each country's government to invest huge money, as Rod suggested, $9 billion US dollars I think this is huge money. So it's not a big money in the world economy but it's really huge money for the basic research. So I think we have to think about why we need so much money. Can we focus on some area to use less money to achieve more? As Qifan mentioned this morning, I really interesting on his suggestion a few years ago, try to generate the super green rice. Because of this, can use if we could use less input and get more yielding, and even better for environment, we needed to integrate the elite variety, the worldwide together, then to share this, the material together. Finally, I would like to say, in this kind of the meeting, we have to let some people work on the gene function, gene cloning, and functional analysis, and also for the work on the gene regulation mechanism. Because this work, this individual, the research is still important for rice functional study, because this is more challenging and more difficult, sometimes, for some. So I think even for a Arabidopsis community, recently I've been in the meeting in Vienna. I found so they focus on most of the basic as plant researches, the questions. But for rice, I think we can focus on the trait, because we have so many varieties, so many cultivars we can use. We can treat this variety as a natural variation. But for the Arabidopsis, they tried very hard to collect so many equal types, But for the rice community, we can focus on the natural variations collected in different areas, and also combined with some mutant libraries. So we could achieve better. Okay, thank you.
Thank you very much. Nori.
So, good afternoon. I myself are interested in the hybrid rice and the hybrid sterile phenotypes. But in the rice community in Japan, I am responsible for the project of the National Bioresource Project, and dealing with wild rices and the chemical mutants and the integration lines from the wild rices into the cultivated rices. So this is the 11th year of the National Bioresource Project in Japan. So thanks to the support of the project, we could manage much information about the wild rices and the other resources. So that is a reason why we could collaborate with Binhans and as and wide relatives of rices. And but using the wild rices, just like as it is and project, we are interested in the integration of very useful genes into the cultivated rices. So several groups are trying to generate such lines. So we hope we will be able to release those resources very near futures. And I think it's very important to settle the universal common integration lines, or something like those lines, which are very useful to use every researchers and the breeders to test their characters and the targets. And as well as it's almost also very important to list up a very specific target in the specific regions in the world to target the demand of the farmers and the breeders and the researchers and genes. So, and parallel progress of those and the efforts will be very important for all of our collaborations in the near future 2020 activities. It's mine. Yeah.
Great, thank you. So I think we've heard some very diverse viewpoints representing our diverse areas of expertise here. And of course, we don't claim to have the full breadth of expertise, but we've heard everything about, or we've heard mention the importance of mutant resources and funding for those, and epigenetics and systems biology, plant breeding and the technologies to advance that, production systems, C4 technology, community focus, germplasm sharing, data integration, importance on focusing in rice on traits, hybrid rice, hybrid sterility, accessing the diversity in rhiza germplasm. So many different ideas. So I guess one challenge will be, how are we going to prioritize these very diverse ideas when a community is made up of investigators? So now I'd like to turn the floor out to you, the audience, and ask what are some of the issues that we're missing here? What have we not mentioned? And what are your ideas for how we might prioritize these different challenges? We have some brave souls who are willing to stand up and present a few ideas that we're missing. There's one. Excellent.
I'm Dr. Jawahar Ali. I've come from Erie. I'm a plant breeder. My main concern is how this functional genomics can help a plant breeder in the first place. Because all this we have been seeing for last several years after the first genome was sequenced. And till now we don't have any kind of tool that can be useful in the hands of the plant breeders. Still the plant breeders are using their own age-old methods, using different approaches to breed better materials. How this functional genomics can be integrated into the plant breeding that we'll see in the coming decades. Thank you.
We bridge that divide. Do we have any responses, either from the distinguished panel or members of the audience? I know it's always a challenge to get breeders talking to molecular biologists, and usually molecular biologists tend to be very reductionist, very focused on their gene's function. Genome biologists are maybe focused on applying their tools. Plant breeders have very real-world problems that they need to address. And so how can we make the information that we're gathering from genomics and molecular biology translate better to plant breeders? Antonio.
I'll try. I think we have several examples of the use of genomic tools in the field. Sub-1A is one example, very nice. And we, as a professor in plant breeding, I take my students to visit breeding programs in Brazil. And I've seen over the years an increase in use of marker technologies, SNP technologies in big companies such as Pioneer and Monsanto, RiceTech. So I think we have seen steady progress of the use of these technologies, and this comes from the translation of what we produce here into the more applied breeding. Maybe I tried to answer that, and I have one point is that we need to also point out that all the problems we're facing with rice now, and we have to add the changes in the environmental factors that will maybe harm our future harvests. So we need to be able to make this awareness go to the governments and say that, okay, we are doing fine with such a steady progress in gain, but if these environmental factors continue to change, there will be a dynamic process, and we need to be prepared to have genes that will be useful in making plants more resilient to these environmental changes. That's fine.
Very good. So, building the tools for a better plant tomorrow, and we have a response.
Yeah, I do agree that the marker-assisted selection and all those simple tools already have been integrated into plant breeding. Plant breeding is already grown to that level where marker-assisted selection is already being incorporated. But what we need is the sequencing. We are talking of the sequencing technologies that we talk about 3K, we talk about 10,000 germplasm sequence information, which will be remaining in many of these databases. How best we can utilize this kind of information in the coming decades. That's what is my challenge to the functional genomics guys. Okay, here is a set of genes. If you put them, okay, the potential will increase. How that can be integrated into plant breeding. Plant breeding is already grown. We know that marker-assisted selection is a part of plant breeding.
Okay. I think because the resequencing now is so powerful to detect the polymorphism in each different germplasm. I think now it's time to collect enough the elite varieties and try to do the sequencing to determine the polymorphism. To detect the polymorphism, to try to identify how many elite traits or elite genes have been used for breeding. But we know because in rice there is a strong linkage disequilibrium, because some elite trait could be strongly associated with linked with some the better the genes. And but nowadays the people we are working on the GWAS, we try to use a very diverse sample. But if we can use some of the control design case design study, and we definitely can identify the most of the the elite the trait the people use it for breeding. Then we can provide this information for breeder, to let them know what kind of the germplasm can be used for your breeding. But in my lab, we are working with some other rice breeder in China. We already sequenced more than 800 elite variety. We named some, we give some definition to name the elite variety. But many years ago, the rice breeder never believed, the molecular genetics, the publications, and if some gene could be increase the grain size, the grain weight. But for the breeder, they need more than 1,000 elite, the trait can be, to breed, a better varieties. So I think now it's time to integrate all the information. And another way, it's time to identify allelic variation on some of the important locus or loci. But those information also can provide it to the breeder to use it, for breeding the new varieties.
Great. So a shared love of DNA sequence data can bring together the genome scientists and the breeders, perhaps. Yes, Jifa.
Come back to Li Johor's question. So I think a couple of components are very, very important. Component number one is the breeders are really interested in these sort of things. So that means a lot of changes in the breeding activity. Like in China, we have thousands, even tens of thousands breeders. They are in very, very small group. So they don't care about the advance of genomic technology. So that is one thing that is very, very critical. So this needs to be some sort of reform to put together to enlarge the team of the breeding teams, or probably big company activities like whatever you may not like, but that's something necessary to get the size of breeding activities bigger, so that they would care for the technology. That is very, very important. That's a fundamental part of the breeding, making use of the information we developed. So number two, I hope you have noticed in one of my slides this morning, I defined what's so-called genomics-based selection or genomics-based breeding. So there are two important components. One is a selection chip, which should incorporate all the information, including the SNPs from the sequencing activity. Whatever number of sequences that should come to a SNP that could be provided to breeders for distribution, for utilization. So the chip should include the diversity, the sequence diversity, at the same time the allelic information of the genes cloned. Whatever the genes, whether this would be very useful, likely useful, or not very useful, but those genes should be the information of the genes. The sequence variation of the genes should be incorporated in the breeding chip. The third part might be we should develop gene-specific selection system. That is also what I presented this morning. Say, for example, we are interested in, probably the whole world would be interested in blast resistance, for example. So we need to develop breeding systems for, if not all, the genes that are useful for improving blast resistance. So this put together, I think the genomic research could offer a very, very good solution to rice breeding.
Very good, thank you. And maybe we should also mention training the next generation of young scientists is critical, because a lot of these the incorporation of new approaches comes from the young people in your group who don't understand barriers so well, and so are free to work in, whether it's informatics or in the lab or in the field. And so making sure that we train young scientists and get young scientists interested in the problems that we face, I think, is also a pretty critical step to make sure that this is carried on. Okay, so do we have other members of the audience? Yes, the back.
Okay, I would just like to add on to this as a young scientist. So how would you encourage young people actually to work in rice, especially if you're from like the Western world where rice is not really a big topic, but still a lot of the funding comes from these countries. So how would you like encourage people to work on this, and how would you like lower the entry level coming from a different crop, like plant, for example, Arabidopsis? It's very difficult to change into rice because there's no information resource available where you can just go and look up, or like it's very difficult. And the other point I would like to raise was there are a lot of community efforts made, which I think are great, but how do you combine this with young scientists who need papers to get jobs afterwards? How would you like to incorporate this?
Yeah, very good question. So how can we encourage young scientists? And the question of papers, allowing people to advance their careers in genomics, I think, is also a big challenge, because so much of what we do is team-oriented, and developing some of the resources, genetic resources, whether it's populations, mutant populations, mutant collections, characterizing those. It can be a real challenge if you have a team of students working on that, where each student needs to have an individual success in order to advance to the next generation. What can we do to help those people to develop their own research projects and questions? So we have ideas from our distinguished panel, or the members of the audience. Sorry. All right, so I would also throw out a few other ideas. Maybe issues, there's social issues that we need to deal with. Acceptance of genetically modified crops. I don't know that GM rice is that widely used in the U.S. I don't think we have any genetically modified rice that's commercially produced. And so I think we also really need, as scientists, we really need to reach out to our communities and talk about the issues, the benefits of genetically modified crops, the genes that we work on and the reasons that we work on them, the traits that we work on them, or are we going to have to wait for some crisis to occur by which it's really the only solution is a genetically modified organism? You think about something like papayas in Hawaii, where it took the devastating ring spot disease in order for the genetically modified papaya to be accepted. Okay, so do we have other points that we want to raise related to grand challenges and rice and how to address the next green revolution? Yes, we have another bold soul at the back there.
Yeah, Sigrid Heuer from Erie. Based on our experience at Erie, is what we find that often the genes that are really high impact, like SUB1 or PUB1, or now that we find drought genes, they're not necessarily coming from varieties like Nippon Barley. And whenever I read the papers or the beautiful work that is being done in research, and I see it's again Nippon Barley that was used, I wonder if we switch to other genotypes that we know are tolerant of multiple stresses, for instance, and do all the science using those materials. I think then we could advance much faster in our research and make much better use of the genomic tools that we have.
Great, thank you. All right, we have Philippe in the front here.
From Biocorp Science. Just maybe a reflection that this community should have is about the interaction between your community and the rice industry. The industry is now investing in rice, but it's not an easy business. So it's an important transition at this time, where we see the opportunities, the challenges. At the same time, you should understand the real positive role that the industry will play and try to identify the areas where this community can really partner with the industry. Because if we don't make that work in the next 5 years, the industry may simply plug the play, because the business is not sound enough, and that would have a very negative impact to achieve any goal we have talked about today. So, and probably the rice community has to learn the lesson from the maize community or other crop, where somehow at the end of the day the private-public partnership is very beneficial. Also at the beginning, during the early stage, it has been perceived maybe as a bit painful. But this transition for rice should be much smoother if we learn from the other crop. So that would be nice maybe to hear feedback of this group, or any question about experience of people who have been working with the private industry or people who have been working on both sides. That should be a key point on the agenda, I think.
Right. No, I think that's a very important point. I mean, there definitely are major differences between maize and rice in terms of the people that are growing them, the economies and the social implications of the two. But clearly, in industry and the impact on maize has been tremendous, and it's really a driver of technology and the application of technology and the use of germplasm. So I think it would be interesting to hear what people think about the potential for tapping industrial know-how approaches for facilitating the contributions of industry to driving the next green revolution in rice. What is the potential? Does anyone have any experience with that they could share? Probably most of us are publicly funded and predominantly public projects, so I don't know, Philippe, maybe you have specific ideas about what it would take to get a company interested in contributing to public projects, and what can we do as rice biologists to entice industry to devote more resources to rice research?
Okay, maybe I make one or two comments. Very often, by many institutes, the rice industry is seen as a few players: the Bayer, the Syngenta, the DuPont Pioneer. We are big players because we are in multiple crops, but locally there are also big players. There are big players coming from China, and I'm sure Chinese institute will have more and more interaction between domestic companies in China for their project. And in the future, you will see those companies in China more and more visible for the rice community outside of China. It's probably not the case today. So don't necessarily. And in other countries, there are also local players which are very important. Also, they may not have the resources to fund any project. On the other hand, there are many funding agencies, including the Bill and Melinda Gates Foundation, now approaching the rice industry to hear our perspective. Because, of course, they have started to go to the place where the knowledge is. So in the rice community, they immediately go to the public institute, because rice, including rice breeding, has happened in the public community. But of course, because of the involvement of the rice industry, we are being consulted on where we think the right investment can be. And of course, we always have an holistic view. We don't necessarily look at the trait per se. We look at the priority of the trait compared to other technologies. And those other technologies you may not be fully aware of, but they range from a seed treatment technology, which can address a part of the cycle of the crop, or they may involve biologicals application, synthetic chemistry application. So I've heard about BLAST. I think we have also an interest in BLAST, disease resistance, for the long-term perspective. We see that as a key opportunity. Now, for the next 10 years, we believe we can provide farmers with solutions for blast tolerance. So that's how the industry, when you engage this dialogue, can help you prioritizing certain traits. And I also, we have communication with IRRI. All the big players talk with IRRI. Now, the dialogue with IRRI, to be a bit provocative, has evolved in a direction which does surprise the industry. The mandate of IRRI, like other public institutes, is open sharing. And now, when we approach IRRI, we get a kind of feedback that is, we need a confidentiality agreement. We were approached to have a blanket confidentiality agreement, to which a request I reply, if on behalf of Bayer Crop Science I sign for the first time ever a confidentiality agreement for any discussion we may have in the future with any scientist at IRRI, I will ask IRRI to make this confidentiality agreement signed by any partner of IRRI. So we have to be very cautious in the public sector not to resist to the industry. So don't as a public community, don't send the message to the industry that partnership will be difficult, because experience in other crops shows that this is exactly the opposite. So, of course, the industry will always come with some objectives which don't necessarily address the entire rice growing countries. So we will focus today mainly on hybrid rice technology, and we will prioritize our trades towards this objective. In China, we are in talk with some companies who are also showing strong interest in inbred technology. So again, China is a very particular case where a lot of things may happen, and the rice community, I think, in other countries will learn from the private-public partnering model happening in China. So maybe Chief or any other person in the audience may comment on that, but I think that would be a very important to watch in the next probably 2-3 years, what's happening in the rice industry and public-private in China.
Great, thank you, Philippe. I think we also need to make sure that the public collaborations are as open and as flexible as possible too, so that we can have good collaborations internationally. So maybe I'll mention now, maybe we can wrap up here, but there's another room that's available for extended discussion right after this, so we'll maybe just take a moment for any closing remarks. It's the Ban Pun Ton room. Sorry for my tie, I think that means rice is awesome. That's on the 6th floor of level 2. So anyone who's interested to continue this discussion, maybe in a smaller format, a little more intimate discussion, why don't we retire to that room and just take a couple minutes break, stretch your legs, and then we can head over to that room. Again, that's the Ban Pun Ton room, which is in level 2 of the 6th floor, the floor that we're on. So maybe we just take a minute. Anyone have any last remarks that they want to make before we retire to the other room? We're all tired, some of us jet-lagged. All right, so with that, I'd like to thank the members of the panel who came up here, and thank members of the audience for your participation as well. So we'll see you in the next room in a couple minutes if you're interested to continue the discussions. Thank you.